motto of this particular organization is that your local window to global opportunities in computational biology. The structure of the ISCB Student Council is such that ISCB-SC is an international organization founded in the US in 2006. The Student Council is exclusively composed of students from across the globe, fostering young researchers and early career researchers and their engagement in computational biology. RSG India is regional student group composed exclusively of students from Indian educational institutes under the Student Council. So the ISCB was founded in 2006. It nurtures the next generation of computational biologists with RSGs in over 26 countries. RSG India's purpose is to connect and promote a strong nationwide network of computational biologists, linking local talent to global opportunities. So there are, as mentioned, there are over 30 RSGs active across the globe in over 26 countries, one of them being India. And under the Indian Regional Student Group, we proudly collaborate with premier institutes, including Mahindra University in the Hyderabad, NIT Calicut, and Manipur Academy of Higher Education in Karnataka, as well as the Tamil Nadu Node, which is Sri Ramachandra Institute of Higher Education and Research, which is where we are all from. Here is the RSG India Core Committee. The board is composed of members for the year 2025 to 2026, supported by faculty advisors and visionary leaders across India. Professor Manish Kumar is based in the Department of Biophysics, the University of Delhi South Campus. RSG India has successfully organized impactful events in the past, fostering collaboration, learning, and innovation in computational biology. These are screenshots from the various events conducted in the year 2024 and before. So we look forward to hosting more such events and expect your active participation and contributions. You can reach us through our website or connect with us on social media platforms. I would now like to call on the Tamil Nadu Node Secretary, Syed Abdul Aziz, to introduce the Tamil Nadu Node and the purpose and mission of our team.
Good evening, everyone, and a very warm welcome to everyone who joined us this evening. I am Syed Abdul Aziz, and I'd like to take a moment to introduce the wonderful team that is the Tamil Nadu Node of ISCB RSG India. We are a student-driven community established in the year 2025 with a vision of creating a vibrant platform for aspiring computational biologists and bioinformaticians. Our goal is to create a platform where bright minds can collaborate with researchers and professionals through events, workshops, and webinars, thus encouraging learning and collaborations. Next, I'd like to introduce the team behind this effort, consisting of members from both undergraduate and postgraduate level across all these years. Here is a photo from our very first event, on-campus event held at SRIHER. Established in 2003, our Department of Bioinformatics at Sri Ramachandra Institute of Higher Education and Research offers both BSc and an advanced MSc course in bioinformatics, with a comprehensive curriculum blending the basics of biology and technology. We focus on hands-on learning, interdisciplinary approaches, while being on par with the latest developments in computational approaches in life sciences. Our students graduating from this department have gone on to make a difference in leading companies in the industry across the globe, such as Pfizer, EMBL, Nucleum, Biocon, etc., and also in academia with institutions such as University of Cambridge and University of Toronto, TUM in Germany, etc. And we are also proud to mention that we've previously had many of such distinguished alumni on our platform as guest speakers to share their valuable insights. And a special mention to our advisory board of our committee, consisting of our faculty, Dr. Dicky John Davis, Dr. Abhinand PA, and Dr. Arundhati M. And now I'd like to pass it on to Dr. Abhinand to introduce our guest speaker. On to you, sir.
Thank you, Abdul. It's a proud privilege for me to introduce the speaker of the day, Dr. Dicky John Davis, sir. I'm really proud to say that I'm one of his students who studied under him from 2008 to 2010 when I was a master's student at the Department of Bioinformatics at Sri Ramachandra Institute of Higher Education and Research. Sir has over 22 years of teaching experience, and he holds a PhD in bioinformatics and has multidisciplinary training in the fields of molecular biology, biotechnology, and computational biology. He pursued his PhD, and before that, he has a MPhil in biotechnology and also has a master's in molecular biology. He has several years of research experience in domains such as metagenomics, transcriptomics, molecular modeling, and also in development of databases related to biological compounds. Sir has published several high-impact papers. Almost 30 papers he has published so far. And one of his main contributions to the field of bioinformatics is the development of the seaweed metabolite database, which is actively being updated. And it is one of those databases that is actively used by several researchers who are working in this domain currently. And he has been a invited speaker at several national and international conferences, and he has also authored 2 book chapters and made several submissions to CBI GenBank also. So we are very proud to have Sir as a speaker here, and he's an invited journal reviewer in many of the international journals, including PLOS One, PLOS Computational Biology, Journal of Biomolecular Structure and Dynamics. So we are very eager to listen to you, sir. It's over to you, sir. Thank you.
Thank you, Abhinand, for that nice introduction. It is first I thank the organizers for inviting me, and also I send my warm greetings to all those participants who have come here. Shall I share my screen, Harshidi? Okay, yeah.
Yes, sir, in a moment. And with that, I would like to remind everyone to stay until the very end to fill out the feedback form that will be shared in the chat at the end of the session to receive your e-certificates. Thank you. Over to you, sir.
Yes. Okay, 1 minute, just a minute. I will just. Okay. Good evening, everyone. And today I will be talking on the identification of potent marine algal compounds, where I'll be talking on the database that has been created and using the database, QSAR studies that has been done on that, and molecular docking of the compounds, further molecular dynamic simulation, and MM-GBSA is what I'll be talking about on today's session. And I already have a lot of questions from your side, and I have tried to incorporate some of the answers to the questions in my PPT. And if I have not answered your questions. You can always post your questions in the chat. So with this small thing, I'll just start my presentation. Right. So so hope you are all able to see the change in screen. Okay, fine. Right. Okay. Marine algae is one of the ocean's rich source of biological diversity because they live in a stressful and diverse marine environment, and this helps them to create compounds of a variety of chemical diversity. So, because of the salinity they have, and because of the predators that eat the marine algae, you have a lot of compounds that are secreted by them, which are of varying chemical diversity. And the importance of marine algae is that about 90% of the species of marine plants are algae, and about 50% of the global photosynthesis globally is derived from algal only. So, and algae is a renewable source, and every you can cultivate, even cultivation of algae is also there. So, seaweeds are marine macroalgae found in the intertidal region and in the seabed. And some of you asked what is the use of this marine algae or the seaweeds. They are used in agriculture as natural fertilizers, soil conditioning, plants biostimulants for growth, boosting growth, yield, and stress resistance by applying nutrients and hormones, improving soil structure, and enhancing water retention, all while being a suitable alternative to the synthetic input. So this is one of the main advantage of uh, having uh, main advantages of having this main advantages of having this marine algae. Here I have also given some of the, I'm not marketing these, these are seaweed extracts which have been used as antioxidants for thyroid support, and you also have them for uh, as for gut digestion. You also have these also for um, other things like nutrients also for thyroid. Also you have these supplements of algae. This is a brown algae turbinaria. They are classified based on the pigment they process. So this is turbinaria, this is a red algae laurentia, and this is a green algae, and this is um, it's a powerful plant booster, which is also available. All these are things which are available online. So, with this short introduction of what is marine algae and the importance of marine algae, I will go into the database that was designed. So, basically, chemoinformatics focuses on extracting, processing, and extrapolating meaningful data from chemical structures for the purpose of making better decisions faster in the area of drug lead identification and optimization. So the database, which was designed by me, is called as seaweed metabolite database, because when I was working on these seaweeds, I found that there are a lot of compounds or secondary metabolites which are secreted by these seaweeds. Basically, the primary metabolites are alginates. Uh, all these are the primary agar, alginates, fucoids. All these are the carbohydrates which are the primary minerals. Apart from that, they have a lot of secondary metabolites also. The and here this database has around 1077 seaweed compounds in this. So you can search the database using the accession number or the compound type or using the binomial name or by the IUPAC names of the compounds or by wild characters also are supported in this. And in this database, you will find the general information of what is the compound and the molecular weight and monoisotopic mass. Those are general information. You'll have external links to PubChem and ChemSpider, and you have the seaweed information. What is from which organism the compound is there, and most important is that the compound would vary from one region to another, one season to another. The metabolites would vary. So here I have also documented from which origin or geographical origin the seaweed is from. And the other one is that the biological activity, what is the extraction and what is the biological activity, because this biological activity only helped me to go on to the next thing on QSAR. So you have the biological activity. Apart from that, you have the structural information of the IUPAC names, Miles notation, and in Chi you also have predicted properties of the compounds and the references. Here I would highlight here that this database was created from literature mining and data mining of journals from 1965 till date. I have extracted literatures from them and have put the compounds in this database. So this database has presently 1077 compounds, and mostly it is red algae, and it is mostly dominated by the red algae laurentia, which has 542 compounds along with the other compounds. Then you have the brown algae, which is 246 compounds from the brown algae, and then the green algae. All these are you can find in the database, and you can also download the compounds as either as a PDB file or a SDF file. You can download the compounds, and this was published in Bioinformation. The next thing I'm going to talk is of structure, quantitative structure activity relationship. So QSAR is a method where it correlates measurable and calculable physical or molecular properties to some biological terms, activity in terms of equation. So basically, you have the structure of the compound, and from the molecular structure of the compound, you represented it, and from that you take the descriptors, you calculate the descriptors from it, and you model it using any machine learning languages. You model it and you equate that to the biological activity. So this is what is QSAR. So the activity is equal to the function of the molecular descriptors. So the success of a good QSAR is what is the right descriptors that you are going to take, and what is the right method or what methods that you are going to do for machine learning methods like regression or correlation or partial least square analysis, whichever method, the right method that you're going. Based on that, you're going to get a successful QSAR. So once you have a valid QSAR, it is possible to predict the biological activity of related drug candidates. So that is the highlight of QSAR. For unknown compounds, you will be able to predict the biological with for because you with known compounds you try to have the biological activity, then you can predict for unknown compounds. So QSAR should be accumulative, that is from the structures with the activity data you should be able to tell what is what that is present in the structures is responsible for the biological activity. I think I will demonstrate this one. And it should also be predictive, is that you should be able to tell the uh, from the structures you should be able to tell the without the activity data you should be able to tell what is the activity of the compounds. So it should the QSR should be both explanatory and also it should be predictive. So using QSR, you can explain the localized properties of molecules such as the binding properties, whole molecule properties such as uptake and distribution also you can. So this QSR is applied in various fields in both in chemistry, pharmacology, toxicology, drug discovery to predict the biological activity and the physiochemical properties or for toxicity of chemical compounds based on the molecular structures. It's not only used in any chemical industry, QSAR can be used, but the method is almost the same. Only thing you should have the physical or the chemical property of the compound. So QSAR relies on the principle that similar molecular structures often exhibit similar biological activities or properties by analyzing a data set of known compounds with measured activities. So the mathematical methods you can use, mathematical model you can use regression or classification to build to correlate the structural features and represent the molecular descriptors such as with the target response such as the potency against receptor or the toxicity endpoint or the solubility. Now you have some free tools for this QSAR. One of the free tool is this RD kit tool. This is based on Python libraries. Uh, it is used for chemoinformatics. So it is a free tool. You can do different types of QSAR using this RD kit. Apart from this, you have also commercial ones like PipFlop or Deep Auto QSAR, which is from Schrodinger. And we do have other tools also which is available online. Um, but this as a Python now it is more easy to use using this RDK. And there are different types of descriptors, how you calculate this based on that. We have 2D descriptors which uses the 2D structures of the compounds like the fragments and the topological indices you'll be able to create using from that you'll be able to do the 2D QSR. On the other hand, 3D QSR incorporates 3D molecular fields like the molecular comparative molecular field analysis or COMFA, analyzing steric and electrostatic interactions. And you also have higher dimensional QSAR where it includes 4D to 60 variants accounting for the conformation or dynamics. So you also have group fragment based or GQSAR which focus on specific molecular fragments. So this is a brief introduction of what is QSAR, what tools are there, how do you create the descriptors on that. So in my present study, I used the compounds that were there in the CV data, metabolite database. It has around 1077 compounds and you have it from different red, green, and brown algae. And in that, as I showed you in the database, I have incorporated the cytotoxic activity or the anti-cancer activity of against nearly 43 different cell lines. So, and I have used machine learning to understand and exploit the relationship between the chemical structures and the biological activity. So, uh, though there are 43 different cell lines, I have just, for the study, I have just taken 6 cell lines because all these 6 cell lines have more than 40 compounds in that. So from these are different cell lines and these cell lines, what cancer type it is, and the number of compounds that is there in the database listed down here. So this is the diverse structures or cytotoxic compounds in the CV metabolite database. You see different structures in these databases. So where the 2D and 3D QSAR will take these structures into consideration and make descriptors from these structures. So the molecular descriptors or the chemical features were extracted from molecular structures using VLife MDS. So nearly we had the 239 descriptors was calculated, and which includes physiochemical properties or 391 alignment independent descriptors. We also have biological descriptors or relevant uh, cytotoxic activity, which is in the inhibitor, that is the IC50 values of the compounds. So this is the distribution of the IC50 values among the cell lines. Yeah, so these are the cell lines, and what is the number of compounds in each of it, and the distribution of the IC50 values among the cell lines. So I have developed the model using the linear forward selection. Actually, here I have used the software called as Vega, for Vega, which stands Vigat Environment for Knowledge Analysis. This is also a free tool where I use the correlation-based future selection algorithm that is in Vega. And for selection, I use the hybrid optimization technique where genetic algorithms were used for searching the descriptor space, and multiple linear regression was used for fitness of the evaluation. So for all the 6 cell lines, I tried to find the correlation of the descriptors, and I found that after 3 descriptors, you see that you have a plateau here. So with 3 descriptors, I think that I can calculate the multiple linear regression for this one. So based on this, um, basically the methodology that was adopted was I've taken all the compounds in the seaweed metabolite database, and so these are the 43 cell lines for which I had the IC50 values of different compounds. And so the anti-cancer activity I had for 245 compounds, and out of that I have chosen only 157 compounds against 6 different cell lines that I've taken, and then I've calculated the descriptors using VLife MDS, where you have the topological, geometrical, and hybrid repertoire. The other important thing why I use this VLife MDS is that you have government's alignment in independent descriptors, and dependent variable was the PIC50. And then I use Vega for division of the training and the test set. And for generation of the QSAR model, I use the linear regression classification along with the genetic search to classify the descriptors and the study. And then for prediction also, I had a 2 test and training set for each of it, and I validate the QSAR using the lever node, average residue, and the Fisher statistics. I try to find out which is the best statistically significant ones. So R square is the square of the correlation coefficient, will reveal the degree of influence of the corresponding descriptors on the target property. So square of correlation constant greater than 0.7 was inferred as significant. And for internal validation, I use the leave one out method, where each molecule in the training set was eliminated once, and the activity of the eliminated molecule was predicted using the model developed by the remaining molecules, and the leave one out method, anything greater than zero point 0.5 was inferred as significant. And for external validation, I used a test set that was not used in the training set, and the activity of each molecular test bed was predicted by using the model. And here I used two independent test set for constructing and to rule out the chance correlation. And average residue or the absolute difference between the experimental and the predicted values, the lower the average residue, the more significant was the model. So, so this is the training and the test set that I've got. So two tests that I have done for training, and both were independent training and test sets. And this is the regression equation, the summary that is also for all the 6 cell lines. I classified them into training set, test set, and the predicted data set. And you add some outliers. I removed those outliers. And this is the regression equation for each of the cell lines. So this is the regression equation, and the correlation coefficient is R2, the correlation. Anything more than zero point 0.7 was taken as significant, and the cross validation or Q2, anything more than 0.5 are taken as significant. And average residue also, the lower the average residue, the better is the stats. And I have based also have done the Fisher statistics that measures the ratio between the explained and unexplained variance of the for the given number of degrees of freedom. The Fisher statistics were also calculated. So higher the F test, the more significant is the model. So it's a regression summary. So based on this, all these models did well. Uh, and based on the descriptors that you find in this regression equation, I put as a pie chart where you see that most of the descriptors were alignment independent descriptors. The importance in this alignment independent descriptors is that these descriptors actually contributed for the cytotoxic activity on the cell lines, because most of the seaweed compounds had either bromine or chlorine in their thing. So the distance between the uh, chlorine and the bromine in the structures mattered more for the cytotoxic activity. So that is what this descriptors. So here I have developed 22 descriptors, which have 14 physiochemical and 8 Bowman's alignment descriptors. The intercorrelation that appeared in all developed and were taken into account, and descriptors are found to be reasonably orthogonal. And this is the distributor contribution with the activity variation in the 6 different sublines. So some had positive correlation, some had negative correlation, both of it. And so 6 QSAR models for 6 different cell lines with 157 compounds from seaweed metabolite database were built to access the predictive power of QSAR models for anti-cancer activity. So cell lines HeLa and MCR7 showed good statistical quantity where you have an correlation point nearly 0.75 and cross correlation nearly 0.65, followed by A431 and HT29 and 38. This also had a reasonable statistical values. The study reiterates that the cytotoxic activity of the seaweed metabolites is due to the halogenation of the compounds followed by the cyclic ring-based descriptors. These features can thus be used to design and synthesize new compounds with good potency and specificity. So this is one thing that came out from this research, and I published this in European Journal of Pharmaceutical Sciences. So the next thing that I'm going to talk about is on molecular docking. So, molecular docking or protein ligand docking uses the 3D structures of the biological target here, which is the protein, to design or optimize small molecule ligands that bind with high affinity and selectivity. So, the components of molecular docking, all molecular docking softwares have two algorithms that run in common. One is the search algorithm, the other is the scoring function. The search algorithm explores the conformational space because you're going to give a binding site. So the search algorithm will explore the conformational space. So you have rigid docking where you treat both the protein and ligand, you can treat it as fixed, while most of the softwares use flexible docking, which allows only for ligands, and mostly the proteins are as rigid to it. And you have different methods for the search algorithm, which includes genetic algorithms, Monte Carlo simulations, or incremental constructions. All these are algorithms that are used for the search algorithm. And for the scoring function, this evaluates for each post that has been given from the search algorithm. It evaluates the empirical force field knowledge based metrics, and it tries to find the binding free approximation like the delta G will help to rank the courses. So the all our softwares have both the search algorithm and the scoring function. That's where the different softwares vary. So in Autodoc Vina, this is a free software. So it has iterated local search, and it does random perturbations, and it uses empirical and force fields, and as told you, it is free and fast, and this is widely used. You have the gold software from CCDZ. It uses genetic algorithm, and it uses the empirical scoring function, and you have one thing called as the glide score, and this also gives high accuracy for flexible docking. And Schrodinger software has a software called as Glide, a module called as Glide, where it uses hierarchical systemic search and Monte Carlo, and here also it has a Glide score. And you have DiffTalk, this uses differential generative models. And the other one that is web interface is called as the Swiss Talk. This is also now it has two modes where you have attractive cavities, and also Autodoc Vina is now also introduced, is now it has been put into Swiss Talk. And here it uses charm force fields or Vina force fields as same as Vina. And this is a user-friendly web interface. You can do the docking. So because some of you asked the best way to learn docking, you can use this Swiss dock. You don't have to install Autodoc or anything. You can do it as a web interface using the Swiss dock. Right. So for what have I done docking? First, I told that for docking you need a macromolecule or a protein. Here for this I have taken pancreatic lipase, which is a primary enzyme responsible for digestion of dietary triglycerides in small intestine. So in humans, the pancreatic lipase is encoded by the PNLP gene, and it plays a significant role in dietary triglyceride absorption and hydrolysis of triacylglycerols to monacylglycerols and fatty acids. So excessive fat absorption directly contributes to free energy balance and fat storage, leading to obesity. So why I have taken this enzyme is that this enzyme is responsible for fat storage, and also the more fat the stored, it leads to obesity. So the only current medication that is given that is targeted against the pancreatic lipase for weight loss is orlistat. And seaweeds, on the other hand, if your seaweeds are mostly consumed in China and Japan, and you won't see these people who are Chinese people and Japanese people are obese. So mainly they consume these seaweeds, and these seaweeds have a potential of anti-obesity agents due to this their low calorific content and high fiber. You have unique polysaccharides, polyphenols, and compounds that inhibit this digestive enzymes. So uh, so that's why I've taken this obesity and the seaweed compounds. And here for the pancreatic lipase, I have taken it from the protein data bank, RCSP protein data bank, 1 LPB, and have done docking against 1110 seaweed compounds. And also I have taken 126 plant compounds having anti-pancreatic lipase activity, and totally of 126 or 1236 ligands have taken in total to do the docking. Okay, so this is the methodology that has been followed. First, by visual screening using Autodoc Vina, uh, I screened for 1236 compounds, and I kept a filter that those having a binding at less than 0.7, less than 7, -7 were taken, and I had 530 compounds in that. And I did ADME screening using Swiss ADME, which is a free software online tool, and from that I got 443 compounds. Then I did toxicity prediction using Protox to, 2 tool, which is also online. From that, I filtered it to 38 compounds, and then from 38 compounds, I did molecular docking for 5 compounds using Autodoc, and then for these 5 compounds, I did molecular dynamics using Glomax, and also I did MM-GBSA, and I found a lead compound. So these were the compounds, the 5 compounds, the accession number of these 5 compounds. The first 3 compounds were from the seaweed metabolite database. So the first 2 were from the red algae, the third from the brown algae, and these 2 compounds were plant compounds. Epicanthin and this digens is a plant compound that we have taken. All 3 were the lead compounds I have got. And this is the binding energy and the interaction of the 5 best stores along with the control orlistat, which is the standard drug for pancreatic lipase. So here you see that most of the compounds here have a docking binding energy less than -7, unlike orlistat, and you have hydrogen bondings, and you also have hydrophobic interactions and also pi interactions. Ones that I've highlighted as red color ones are the most important interactions which you need hydrogen interactions or hydrophobic interaction is all about. And from the interactions, I did 3D interactions of the ligand. So why I put this image is that all the 5 compounds were docked at the same place, okay, and the interactions of the ligand with the pancreatic lipase, the complexes, okay, and the interactions with it, you have van der Waals, hydrogen bond, and pi donor energies. All these docking energies also you had for these, and these are the ligands and all this that. And in this, you'll see that all 5 ligands had good docking energy, and all their interactions were good. So now I have to find out which one has, because here I'm keeping the protein rigid and only the ligands are flexible. But actually, in when a a ligand comes and interacts with the macromolecular protein. The protein also interacts, and protein is also flexible. So to find the flexibility and also to find which ligand actually fits into the cavity and it's having a continuous activity in the binding cavity, I used molecular dynamics. So molecular dynamics simulation is a method to simulate molecular motion by iterative application of Newton's law of motion, the distance and the direction which the each atom moves in the time interval are calculated using the Newton's law of motion, and the position of each of the atom at time is also being recorded here. So basically, molecular dynamics is that you are calculating the motion of the molecules, and simulation is a mimicry that you are trying to mimic what happens in nature. So that is what is called as molecular dynamics. You try to mimic the behavior of atoms, multi-atom systems in a time-dependent manner, assuming a given potential energy function. So that is what you try to do in molecular dynamic simulation, and the energy function tells about the forces experienced by the atom given the position of the other atoms. So Newton's law can tell us how these forces affect the motion of the atom. So there are different packages which are mostly free, like the GROMAX package, which is Rohingyan machine for chemical simulation. We also have NAMD, which is nanoscale molecular dynamics. You also have CHARM, which is chemistry for at Harvard molecular macromolecular mechanics. We have AMBER, assisted model building and energy refinement. Desmond is a commercial software given by the Schrodinger company. So all these are packages for which you can do molecular simulation. And in all these packages, the MD simulations undergo these steps. One is that you have to generate the topology, you have to define the box, and you have to solvate it with water, where the solvent is mostly water, because in docking we remove the water molecules. Uh, but here we solvate it, so we can see what is the interaction of the ligand with the water molecules also you can do. So you solvate it, you add ions if there are interaction, your sodium and chloride ions you add, and you do an energy minimization, you equilibrate it and you do the MD simulation, and then you do the analysis. So I'm just going to talk about the analysis alone. What do you get after you do the molecular dynamics simulation? So basically, the after you do the molecular dynamics, you get three things. One is the root mean squared deviation. So the root mean squared deviation is that it should be lesser. Okay, that is the deviation from the this. So here you see that BC010, which is a green color, you have the least RMSD deviation when compared to Orlystad, which is the brown color, you have this orangish brown, this is your Orlystad. So BC01, the deviation is less in the RMSD and the pancreatic lipase complexes. Here is a root mean square fluctuation, where based on the residue, what are the residues that are fluctuating or which have an interaction, that you see that the BC010 has less fluctuation when you have interactions with the residues, and the radius of gyration is the other thing which you calculate and which is having a lesser one. Here also you have BC010. So for BC010, the 100 nanoseconds molecular dynamics, the whole video was taken. You see that the compound BC010 actually is inside the cavity throughout the 100 nanoseconds of stimulation. Similarly, here it is Orlystad. You see that This is slightly moving away from the cavity, but it still retains inside the pancreatic lipase. So this is what you get. So based on this molecular dynamics, you calculate the next one, that is the ligand binding affinities using molecular mechanics Poisson-Boltzmann surface area, which because it will take time for the protein to stabilize. So you calculate the ligand binding affinities for the last 10 nanoseconds, 90 to 100 nanoseconds. We calculated for zero point 0.1 nanoseconds time. Here we calculate the binding energy. Here I've done it for all the six molecules that I had, and I found that BC010 had uh, a very good binding energy, electrostatic energies, polar solvation energy, and SASA, also called solvent accessible surface area, and van der Waals. All these are energies will tell you how good the binding affinities for the ligand to the protein tells you. So here I see that PC010 has a very good interaction on that, and this tells you the binding energy based on the residue with the protein. So for the whole protein, which residues are fluctuating in this. Here you see there are, because you have more interaction with this, we have a large fluctuation here in this one, BC010, and you have lesser interactions in orlistat. So this is the MM-GBSA that is done here, and uh, the molecular dynamic simulation studies conclude that BC010 and pancreatic lipase have shown significant binding energy in comparison to plant-based compounds, and also all these start throughout the 100 nanoseconds. So earlier reports have also suggested that marine algal compounds are responsible for inhibition of the lipid oxidation. Thus, the computational approach on pancreatic lipase will illustrate that BC010 is basically 414 dimethoxymethanol. This has a good significant binding energy when compared to others, and this could be a lead compound and belongs to a group of tetraphenyl toluenes from the brown algae Cystoflora fibrosa, and it's a potent inhibitor against the pancreatic lipase. So this study was published in Journal of Molecular Biomolecular Structures and Dynamics. It was being published in that. So I think I have covered most of the topics, and thank you. If you have any questions, you can pose those questions.
Thank you so much, sir. Now we'll open the platform for the attendees to ask any of the questions. If you have any questions, please, you can open your mic and ask the questions directly or post them onto the chat box. And sir, I believe we already have a few questions in the chat box while you were giving your presentation, sir. If you'd like to address them.
So Abdul Munafi is one who has asked, you want to know the files after. Okay, so that you will address that one. Wonderful presentation. Here, Abdul has asked how many phytochemical compounds that you can use, because here you are doing a virtual docking. It is you who have to decide how many compounds you use for docking. So what is the best compounds and the best compounds to bind? So that which has a highest binding affinity, if you are going to use Autodoc Vina, anything lesser than -7 is a good binding affinity. So more than that, you cannot take the compound. That's why I gave a cutoff of less than -7 for the binding affinity. Uh, why Orlistat has low docking scores compared to others, but it is used as a yes. Orlistat had low docking scores as far as when I use this Autodoc Vina. Uh, Basically, Orlistat has an aliphatic chain. So here it showed low docking scores, but when you see the molecular dynamics, you see that Orlistat always stayed inside the cavity and it had interaction with it. So Orlistat, though in the docking score, you had the score was led, but in molecular dynamics, we found that Orlistat stayed in the cavity and it also continually had an interaction in the cavity. That's why we have to check the molecular dynamics. You have to do the molecular dynamics to confirm it. Yeah, so binding energy for metal as asked, that depends what binding energy you're going to choose. Actually, it depends upon what software you are using. Autodoc Vina, the binding energy less than -7 is good. For other softwares, it will differ based on the software that you use because each software has different scoring functions. So that would vary for each of the softwares. Pyrex software, Nitin Suresh has served the docking purpose using Pyrex software. So Pyrex is a good software because in Pyrex also it uses Autodoc Vina only. But the problem in Pyrex is that you have to define the grid box. So defining the grid box is that you should select the residues around the binding site and then you do it. But if you are doing using Autodoc, you can define the grid box easily. So that is the only thing in Pyrex. But Pyrex also uses Autodoc Vina only. Yeah. Any other questions? Okay. Others can post the questions. I would like to address one question that was already asked. So I think you are able to see. Okay. So this was one question by was some of the one of the registrant Venkat Sahiti. So can we do docking with multiple molecules at once to observe the confirmed effect, if any? Yes, you can do multiple molecules at one shot. You can do that. That is called as multiple ligand simultaneous docking. So this would reveal synergetic inhibition, cooperative binding, allosteric modulations, and competitive interactions, and it will also tell you the effect of the inhibitors on the substrate binding. This is multiple ligand simultaneous docking can be done, and this would reveal uh, these combined effects. And this study is usually done for substrate cofactor. Does it have an action against it, or metal ions and ligands? Do they have any interactions? Water mediated binding and combination therapies also. When you want to do multiple inhibitors binding concurrently, you want to do, then you can do this um, multiple ligand simultaneous docking. So there are some studies where they have done this multiple ligand simultaneous docking. So where they have used investigative inhibitor effects on substrate binding enzyme, polyphenol oxidase they've done. And you also have studies on exploring the synergic effect in cancer where they use curcumin analogs and lactabin on the ERP BP4 protein. They identified allosteric crosstalks in SARS-CoV-2 main protease inhibition. You also have on fragment-based drug design where they use multiple fragments that bind and link it into larger compounds. So these are some studies which are available online, though I have not done multiple ligand docking. But you have a tool which is available free. This is called as Moldina. This is also and based on Autodoc Vina only. So, but in this one you can do multiple ligands. I mean, this is a tool which is available free, and this is a paper just publication published this year. Yeah. So the other question by Saradi is there any role of QSAR in docking, and how can we link QSAR and docking? Okay, so linking QSAR and docking, in my study which I showed you, QSAR, after doing QSAR, for that I did docking with protein kinase, where for anti-cancer activity against uh, protein kinase, and I have found a ligand which is from brown algae, calophysin A, which is a potent inhibitor of it. So based on the uh, QSAR, you can do docking also. It can be done. Okay, so my second paper talks on that only, and considering the structural diversity of marine algal metabolites and the limitations of in silico operation, how do you ensure that the selected pancreatic lipase will remain effective and safe when translated? So this is basically in silico prediction. In vitro and in vivo has to be done because uh, uh, molecular docking is just an in silico activity. So most of the drugs, this is basically a screening tool only. So this is better than you screen it in the lab. So that is an advantage of using virtual screening. But in vitro and in vivo studies has to be done to actually prove this one. There's any other if you have any other questions, please let me know.
So sorry, sir, there was a slight disruption. You got disconnected during your previous answer. Could you please repeat it for the audience once?
Which one, ma?
The last question you were addressing.
Okay. The thing is that this is itself virtual screening is docking is basically a virtual screening. So we try to actually predict some compounds in this one. So whichever is effective, and that then only after you do in vitro or in vivo studies, we have to prove that in vivo and in vitro studies, and the preclinical testing is all that. So you have to prove that then only. So uh, we only if it is proven in vitro or in vivo, then only it is correct. The only thing you can, from a large number of compounds, we can screen down to one is what is the best. So that is one thing. So I have another one from Power. As I'm currently working on drug discovery against tuberculosis using phytocompounds. My target protein is 2B35, known as a inhibitor. I want to validate docking protocol proceeding with phytoscreens. Okay, so basically his question is that first thing is that you have to first find for your protein and the ligand is your, so you have not told me which software you are working on. If you are doing Autodoc, Vina or any other software, first you have to do docking of redocking the protein and the ligand that you have in PDB. You have to do that. Here Madhura has asked, can docking scores beyond -14 and -15 should be considered as a scoring artifact? Uh, what software are you using, Madhura?
Sir, like Autodoc.
Autodoc, then this is an artifact.
Okay.
Autodoc maximum you get -7 to -13 is the -14. Beyond that, you see if you have interaction with water molecules.
Okay, sir.
Okay, so that is the main thing. Because in docking you should have removed water molecules. If you have interaction with water molecules, you will have false positive results. So there was also another question: can you use water molecules using docking? Okay, you can retain the water molecules if the interaction um, is through the water molecules. It is interacting, your ligand is interacting through the water molecules to the protein residues, then you can retain the water molecules. Otherwise you have to remove all water molecules when you are doing docking, otherwise it will give false positive results. Okay, that's why in molecular dynamics we solvate it with water to see if water molecules have interactions with the protein and the ligand. Okay, that's why we have to confirm it with molecular dynamics. Okay.
Okay, thank you, sir.
There were some more questions. How strongly do the results of computational methods correlate with wet lab in vitro results, and what is the marginal error? Actually, this question, because dynamics or molecular docking all is or in silico methods, it has to be evaluated with in vitro or in vivo studies. So the marginal error actually depends upon how good, what software you are using, how good you have taken the binding site and the residues. All that will actually determine how the marginal error. There is no gold standard. What will be the marginal? There is no gold standard like that. That is from Gita Singh. To what extent can we depend on simulations for mimicking biological cell happenings? Actually, it is um, really good, at least 50%, if you are able to mimic it correctly and you are able to interpret the results, your RMSD, RMSF, you are able to interpret it, then it is good. And what is the role of water in the binding site? Do you need to include those water molecules while docking? As I told you, water molecules only in docking, we first remove the water molecules, we remove the ions. So, but we can retain water molecules if the interaction of the ligand to the protein is to the water molecules, because you are taking the protein from PDB where you have a co-crystallized ligand. So when you are seeing the interaction of the co-crystallized ligand and the protein, and if there is a ligand which is interacting with the residue, protein residue through the water molecule, then you can retain that water molecule alone. Other water molecules you have to uh, remove it.
Yes, sir. Thank you so much.
Okay. Hope I have answered all your questions. Yeah.
Yes, so participants, that brings us to the end of the session. Thank you, sir, very much for the excellent lecture. I believe such concepts haven't been discussed in such great detail over the internet so far, and this would benefit a lot of students working on their individual projects in bioinformatics. Thank you very much for that excellent presentation, sir.
Thank you. Thank you.
And thank you all for attending. So if I may now direct your attention to the chat box for the feedback form, which you could fill out to access your e-certificates. You could also refer to the QR code on the screen.